requirement for digestion is that as we know that our food consists of different large molecules. They are large molecules and they need to be converted to smaller molecules. Are there as well as in fat molecules consist of different esterification of glycerol with fatty acid, and protein molecules are also the large linear molecules. So the large food molecules cannot pass through the wall of small intestines, and hence for their proper absorption through the GI tract. So they need to be converted to their smaller form, whether it is carbohydrate, proteins, or lipid, because they are in large molecules. Okay, and for proper absorption in through the GI tract, their smaller unit should be formed, and for that purpose, the different digestive enzymes are helping for that conversion. So first, I'm talking about the different digestive enzymes that are found in the GI tract. Okay, generally, the enzymes that are helping for the digestion. So when you talk about enzymes, so the different enzymes are there for selective substrate, we can say. Okay, for example, the digestion begins from the mouth. Okay, in mouth there is saliva, and the enzyme present is ptyalin, which is also known as amylase enzyme. Okay. This is salivary amylase. Old name is ptyalin, and the substrate is starch. So, with the action of that enzyme ptyalin on starch, it forms the dextrins. Okay, the hydrolytic product of this starch will be from different dextrin, limit dextrins. Okay. So after that, gastric juice is there, and when in gastric juice, the enzymes that are available are pepsin, gastric lipase. And the substrate for this pepsin and lipase are protein fats, and they are helping for the digestion of hydrolysis, breakdown of these peptide bonds, and also the this lipase is helping for the digestion of fats, and which causes the hydrolysis of fats into this glycerol and free fatty acids. Generally, talking about fats, it consists of generally triacylglycerol, and for that purpose, the gastric lipase is there for that. And these individual enzymes will be discussed in this particular topic, okay, like digestion of carbohydrate, digestion of lipids, digestion of protein. In that individual topic, it will be discussed. Okay, these are the certain enzymes that are being found in different secretions like saliva, gastric juice, and they are helping for the digestion of starch, protein, fats, like that way. Okay, similarly, coming to the enzymes found in small intestines. Okay, the pancreatic exocrine secretion. Many enzymes are available in these pancreatic exocrine secretions. Okay, and they are helping for the this breakdown of these different large molecules. The lipids, cholesterol storage, alpha amylase are some of the enzymes. And the lipase, as you know, for the lipase, the substrate will be fat and causing the hydrolysis to move the fatty acids also. Action will be the hydrolysis, breaking of their bonds. Okay. Cholesterol storage, the substrate for cholesterol storage is cholesterol. Okay. And it causes the hydrolysis of this cholesterol and fatty acid esters to reach the product of cholesterol, because in cholesterol there are attachment with this fatty acid also, and that bond should be broken with the individual components. Another is alpha amylase. Alpha amylase, this is the we can say the same type of enzyme found in both salivary amylase, and they are responsible for the degradation of this starch, and also the dextrin and limit dextrin or marbles. Okay, means their smaller components will be the final product of this. And now we are talking about this pancreatic exocrine secretions. Means the different exocrine enzymes that are present in the pancreatic secretions. These are some of this, as well as there are others pancreatic. Similarly, the other pancreatic secretions are like as follows: trypsin. Okay. Trypsin, the substrate for trypsin is protein, causes the hydrolysis, and final product will be polypeptides. Chymotrypsin, carboxypeptidase, ribonuclease, elastase. So these are all the enzymes that are present in this pancreatic secretion. Now we are talking about this pancreatic secretion. What are digestive enzymes that are available over there, and what the action they are carried out. Okay, so generally this type of enzymes, they are causing the hydrolytic cleavage of those uh, substrate to give this smaller products. Okay, and similarly, these chymotrypsins, they are providing polypeptides, carboxypeptide amino acids. So these are the final product we can say. Uh, so these are the, for today you have to know just the name of the enzyme, because every we discuss in this particular particular topic, we can say whether it is digestion and absorption of carbohydrate, lipid, or protein, and that elaborative role of these enzymes will be deal. So for today, you are just doing the overview of those enzymes participation in digestion, right? Helping for the digestion. Cross-border enzymes. Okay. Cross-border enzymes. So these are the enzymes generally secreted by cross-border cells of the small intestine, GI tract. Okay. So some of the enzymes that are found in the secretion of these cross-border enzymes are carboxypeptidase, aminopeptidase, dipeptidase. Okay. So by the name peptidase is there, that is responsible for the cause the hydrolysis of these polypeptides. Okay. So when the polypeptides are hydrolytic broken down, their final product will be amino acids. Okay. Although this enterokinase, important enzyme actually it is helping for the trypsinogen to activate. Okay. So directly it does not act on the substrate, rather in another product, trypsinogen, and activates to trypsin. Okay. And this is the active, we can say, enzymes that helps in for the digestion of this polypeptidase. Okay. And peptides, when these bonds are subjected to get hydrolytic breakdown by this trypsin enzyme. Similarly, sucrase, by the name there is sucrase, the substrate will be sucrose, carbohydrate. Now, carbohydrate are generally disaccharides. Actually, disaccharides are generally being broken down at these cross-border enzymes present in that small intestine tract. Okay, and hydrolysis gives the glucose, fructose. Okay, these are the products when this disaccharide sucrose is being hydrolytically broken. So also the different other enzymes are present in this cross-border cells. Okay, they are secreting the different enzymes. Isomaltase, dismaltase, lactase. Right? Also, these are also the enzymes having substrate as a carbohydrate. Okay? And by the name isomaltase, right, it causes the substrate will be maltose or dextrin-like substance. Maltase is maltose, lactose by the name lactase enzyme. Lactose is substrate. Right? So enzyme generally they are ending with ASA, don't confuse, and the substrate will be OSA. Okay. So different other cross-border enzymes are shown here. The isomaltase, right, the substrate will be dextrin. Then gives when they are hydrolytic program, it becomes glucose. Maltase is the enzyme and acting on this maltose to give the hydrolytic product glucose. Right? Nucleotidase, nucleic acid gives the nucleotides. Okay, and similar nucleosidase, they give the product, final product, and purine and pyrimidines when they're with this phosphorylase removing that phosphate part from this nucleotides. Okay, now coming to another important term, zymogens. So I think you have heard this name, zymogens. We have talked about different digestive enzymes that are present in the outer tract, digestive tract, that is responsible for digestion of different substrate, carbohydrate, lipid, protein. And those enzymes will be deal in detail when they are taught about their specific digestion, like carbohydrate, protein, and lipids, their role on that particular topics. So just there is an overview of those available enzymes in the GI tract and their roles. Okay. Now we are talking about the zymogens. Okay, actually the zymogens are synthesized as large inactive precursors, and they are not synthesized in active form. Okay. So the question is why they are not synthesized in active form. If they are synthesized in their active form, they may digest the tissue that's synthesized there. Okay, so being synthesized in its active form, the problem may arise that they may be digesting the same tissue which synthesizes there. Okay, we are in case of acute pancreatitis, where there is premature activation of digestive enzymes by pancreas, there will be auto digestion of pancreas, uh, auto digestion of pancreas, and that may cause the activated phospholipase A2, which converts lecithin to lysolecithin, and that can damage the cell membranes. Okay, so because the digestive enzymes, active digestive enzymes, may be digesting the tissue or cells, the cells which they are synthesizing them. Okay, so for that purpose, to that is called autodigestion, and to prevent that autodigestion, so it becomes necessary for that zymogens to be synthesized in its inactive form. So that about zymogens. Okay, about synthesis of zymogens. Zymogen as well as the proteins which are being subjected for its different target sites. So they have their signal sequence and terminal signal sequence. Okay, so this is important. The end terminal signal sequence means they give the direction where the proteins should be driving to us. So the end terminals contains end terminal of that protein synthesized, newly synthesized proteins, or we can say now in today's case they are the zymogens. So they need to be, for that purpose, they have the signal sequence in their end terminal. Okay, they have the signal sequence in their end terminal, and that gives direction. Okay, for example, through the transcription and translation process, when the enzymes, such enzymes are being synthesized in this raw endoplasmic reticulum. So through this, because they have this signal sequence in this end terminal, means where the proteins are subjected to get into. Okay, so from that synthesized part, it enters towards the Golgi apparatus, and through the vesicles, it comes towards the membrane. Through this vesicle, they are carrying this membrane. There's zymogens towards this membrane side. So due to exocytosis process, now from this newly formed the vesicles, you can say they fuse with the membrane. Okay, first there is fusion with the membrane and causing the exocytosis. Means being synthesized in these cells, now it comes towards the lumen side. Okay, yeah. So and that direction is provided by this signal sequence and terminal signal sequence, and that is helping for this movement of this zymogen, means where they are supposed to come after being synthesized. So after being synthesized of these zymogens, because they are synthesized in inactive form, okay. Now after that, they need to be converted to its active form, and that is being brought about by this proteolytic cleavage. Proteolytic cleavage in the lumen of GIT. Okay. So example of this zymogens is shown here in pepsinogen, trypsinogen, chymotrypsinogen, proelastase, procarboxypeptidase, prophospholipase. So these are the example of proenzymes. Proenzymes or zymogens symptom means they are being synthesized in their inactive form. Okay, so I have already told why the enzymes are necessary to be synthesized in their inactive form, because if they are synthesized in active form, they may able to digest the same cell or tissue which are being synthesized by for them. Okay, so that is called autodigestion, and to prevent that autodigestion or autolysis, they need to be synthesized in their inactive form, and they are called proenzymes or zymogens. Now, after being synthesized in their inactive form, these zymogens, it becomes necessary for their activation. Okay, some when they get the required environment, yeah, after putting in their environment, they may get activated. The other example is shown of this pepsinogen, activation of pepsinogen. Okay. So as you know, pepsinogen are secreted from the stomach, so the chip cells they secrete the pepsinogen. Now for its activation, what is done? Okay, because first it is formed, and after that it comes into the tract. Okay. It comes into the tracts, and that for that activation of pepsinogen, it is activated by proteolytic removal of 42 amino acids from end terminal. Okay, so because it is protein, large amino acids are there, and for that activation, the pepsinogen is activated by proteolytic removal of 42 amino acids from its end terminal, and that is helping by this HCl. Okay, so once that certain part of that long proteins are being removed, they become active. Okay, so the reaction takes place at pH values below 5, and optimum pH is 1 to 2. So in this way, the zymogens, pepsinogen get activated. Okay, first after the removal of this amino acid, certain amino acid length is there. That is that when that is removed, then it gets activated. So here is shown the activation of trypsinogen in this slide. Trypsinogen is also zymogens. Now first it converts into the active trypsin by the enzyme enteropeptidase. So to get active by this zymogen to trypsin, trypsinogen to trypsin, the removal of 6 terminal amino acid is required. Once that 6 terminal amino acid is required, is removed, then it gets converted into trypsin. So this is the active form of trypsinogen. Okay, means now this can have its active role, catalytic power. Okay, so this is zymogens. So after that, it is being converted to its active form trypsin. Now the role of trypsin is that the proenzymes, other proenzymes like chymotrypsinogen, proelastase, procarboxypeptidase, phospholipase. Okay, so these are the zymogens. Now these are also needed to be converted to their active form, and for that the trypsin is helping convert it into active form. Chymotrypsin, elastase, carboxypeptidase, phospholipase. Okay, so after their conversion to active form, now they can have its role in their digestion. Okay, these enzymes. So first there is the role of trypsinogen to convert trypsin, and there is major role of trypsin to convert all those proenzymes, okay, to its active form. Okay, so I've already told for the conversion of that zymogens or inactive enzymes to its active form, it becomes necessary the certain part, certain terminal and terminal amino acids should be removed or clipped from that part. So after that, it becomes active. Okay, here I've already shown about pepsinogen and trypsinogen. Right? So these are the pepsinogen and trypsinogen. So these are the certain zymogens which need to be get activated. So the strategy is that prevent premature zymogen activation. So how the premature activation of this zymogen is prevented? Okay, so means when in synthesized they are at the point when they are synthesized, they are synthesizing its large form. Okay, in large form for your pepsinogen to get activated, the 42 amino acids should be clipped. Okay, so means if they are not clipped, they remain attached to that pepsin. Means what is doing? It is masking its active site. Okay, the part of enzymes that is active site is being masked, means that overlap, means it is not exposed. Okay, so that extra we can say extra part of that zymogens active site is being masked by certain length of its own fragment. Okay, so in this way their premature activation is prevented at the point of synthesis. Another enzyme inhibitor is the pancreatic secretory trypsin inhibitor, a polypeptide, and that blocks any trypsin that is autonomously activated within the pancreas. By chance, if certain trypsin is get activated in the because pancreas is producing different types of those zymogens or those major zymogens. So if by chance, if the trypsin get activated, trypsinogen is being converted into there is one important pancreatic secretory trypsin inhibitor, PSTI. Right? So that is inhibiting this trypsin molecule, blocks. If it is autonomously activated within the pancreas, by chance, by error, if it is being activated, there is important molecule PSTI, pancreatic secretory trypsin inhibitor. So that will block that trypsin if it is being errorly activated. Okay. So in this way, first zymogen, if they are formed, first they are synthesized in large form. Okay, in large form, because when they are synthesized in large form, their active sites will be matched by certain length. Okay, only when that certain length is been clipped off, then the active site get exposed. Means first to get its activation, that certain parts should be clipped. Then only the active site get exposed, means then it will get activated. Okay, So it must get activated at its point of action, where it has to perform its work. So the what prevents the premature zymogen activation is that first they are synthesized in large form. So when they are synthesizing its large unit, certain part which it is masking the active site that causes premature activation, and another is that by chance if trypsin by error if it gets activated, there is important molecule pancreatic secretory trypsin inhibitor PSTR. It is a small polypeptide that will blocks the trypsin in pancreas, not in other in pancreas. So secretion of this, we can say enzyme digestive enzymes. Generally, there should be regulated means at the time of requirement they should be synthesized in more quantity amount, and when there is not required, when we are in we can say rest phase of this digestion, their secretion should be slowed down. Okay, so there are regulated way. Okay, the regulated way for the secretion of these digestive enzymes, and there is secretagogy. Okay, secretagogy. This actually secretagogy is helping for the secretion of those enzymes or other molecules. Secretagogy are those compounds that is helping for the secretion of the compounds. Okay, here is example of secretagogy: stercoline, stamine, gastrin. Okay, and they are helping for the secretion of this pepsinogen, amylase. Okay, enzymes, pancreatic enzymes, different right, secretins. So generally, they may be neurotransmitter or they may be hormones, and they are secretagogy means they are helping for the secretion of different other enzymes. So secretagogy, what is there? That means this is the substance that causes another substance to be secreted. Okay? Means they are helping for the secretion of other substances. Those are the secretagogy, and they are generally the secretagogies in GI tracts are generally hormones or neurotransmitters. Okay. So example is one with the gastrin. It stimulates proton potassium ATPase in the parietal cells and causes the gastric acid production by the stomach. Okay. Gastrin. So here gastrin is acting as a secretagogy, brings stimulates these ATPase enzymes, okay, in parietal cell, and that causes gastric acid production. Generally, they are increasing the production of gastric acid. So secretagogy, different compound are there as a secretagogy, like there may be neurotransmitter, acetylcholine, histamine, right? And also these hormones, secretin, cholecystokinin. So they are helping for the secretion of other molecules in the GI tract, and that's secretagogy. So some of these secretagogy are neurotransmitters and hormones of GI tract. Okay, they are helping for the secretion of other enzymes that is required for the necessary of digestion. Norepinephrine, acetylcholine, neurotensin, neuropeptide, all these are neurotransmitters. Norepinephrine decreases the motility, increases the contraction of the sphincters, inhibits the secretions. Okay. Acetylcholine increases the motility, relaxes sphincters, stimulates secretions. Okay, acetylcholine. Neurotensin inhibits the release of gastric emptying and acid secretion. Okay. So means they are regulating actually when some are helping for the secretion and some are to certain they try to limit its secretion by inhibiting. Okay. Neuropeptide why? Okay, neurotransmitter, it stimulates the feeding behavior. Right? So hunger behavior, we can say hunger time, that it is determined by the neuropeptide, right? Since it's stimulus, we can say. So some of the examples of secretagogy, hormone gastrin. Okay. So after neurotransmitter, there are certain hormones that is helping for the secretion of other digestive enzymes. Okay, and they are the secretagogy, means helping for the secretions. Now about gastrin, they are released from gastric mucosa and duodenum. The presence of peptides and amino acids. So when they are present in the those GI tracts, they stimulate the secretion of this gastrin. Okay, and ultimately, the gastrin hormone stimulates the secretion of HCl and pepsinogen, and that increases the gastric antral motility. The antral part of this stomach motility is generally increased by this gastrin, and also the increases the large intestine flow. Okay, First, generally we are concerned with the secretion of this HCl and pepsinogen, right? So these are the digestive enzymes. HCl is also strong acid and it also helping for the digestion. First, it itself helps for the degradation of those large molecules. And after that, it helps for the cleaving of this pepsinogen to pepsin. Okay, means is to convert into its active form. Another hormone is secretin. Okay, from duodenal mucosa, secreted from duodenal mucosa in presence of acid in the small intestine. Stimulates the pancreas to secrete water and bicarbonate and also insulin and other pancreatic enzymes. So major role of this secretin hormone is that it stimulates the pancreas to secrete water and bicarbonate. Okay, bicarbonate is also important. Hello, please don't disturb. Okay, we are talking about the different secretory generally of these hormones. Okay, gastrin, secretin. Now coming to secretin, what it stimulates is that it stimulates the pancreas to secrete water and bicarbonate. Okay, bicarbonate is also important compound because it when the food enters towards the gastric stomach, we can say gastric lining, there it encounters with HCl, highly acidic environment. Okay, and that acidic environment should get neutralized, okay, by this alkali, and hence bicarbonate is helping for that neutralization of those acidic component when food enters towards the stomach, towards the low intestine region. Okay, and this is the importance of this bicarbonate. And also this secretin is also helping for the insulin and other pancreatic enzymes for secretion. Cholecystokinin, CCK, short cholecystokinin, proximal, released from proximal in small intestine in presence of peptides, amino acid, fat acid, and it stimulates the pancreas to release the pancreatic enzymes. It stimulates the pancreas to pancreatic enzymes, cholecystokinin. Also, the CCK causes the increased production of hepatic bile. Okay, and hepatic bile and stimulates the contraction of gallbladder, relaxation of this glycerin sponsor OD, resulting in the delivery of bile to the duodenal part of the small intestine. This is the one of the important function of this CCK to cause the stimulation and contraction of gallbladder and relaxation of this sponsor of OD, so that the content of this bile can be delivered towards the small intestine because they are helping for the digestion and absorption of lipids. Okay. Another is also it impedes the pancreas to release the different pancreatic enzymes. So this is the role of cholecystokinin to release the digestive enzymes. GIP, gastric inhibitory polypeptide. Okay. Another important hormone, we can say gastric inhibitory polypeptide. They are released from small intestine in presence of glucose and fat. Okay. And it inhibits the gastrin-stimulated release of gastric acid. Motilin, another important hormone is the motilin. And they are released from the stomach and small and large intestine in presence of biliary and pancreatic secretions. Promotes gastric emptying and increase the GI motility. Motilin. Another hormone is somatostatin, released from stomach, pancreas, upper intestine. And actually, it is inhibitory of this gastric motilin and pancreatic secretion because their secretion should be regulated. Some are favoring for the stimulation and some are to some extent inhibiting. Okay, so that their amount becomes controlled and decrease the motility of GI tract. So there are pancreatic secretions and different bile secretions. Their secretions are important because they are helping for the digestion of all those molecules. So from here we will continue in the next class, pancreatic secretion about those. Okay. So in today's class, I'll just talk briefly from beginning, overview of today's class. So digestive enzymes. So we are talking about digestive enzymes. So and for the digestion of different large molecules, first they have to be converted into small fragments so that their absorption becomes favorable from the GI tract. Okay. For that purpose, different digestive enzymes are present in different locations, from mouth to towards up to small intestine, we can say. The different enzymes has been secreted, and they are secreted, and they can act on different substrate. Okay. So saliva has styolin, also known as amylase. Of amylase, starch. The substrate will be starch. Gastric juice has pepsin, gastric lipase, and important enzymes are secreted through the pancreatic exocrine secretions. Okay, so different. I am repeating the previous what we have learned. Lipase, cholesterol esterase, alpha amylase, right? All those are the enzymes present in pancreatic exocrine secretions. Similarly, trypsin, chymotrypsin, this carboxypeptidase, elastase, also these are the enzymes present in pancreatic secretions, and they are helping for the digestion of protein and different type of nucleic acids. So, causing the hydrolytic breakdown of those compounds. Similarly, the cross-border enzymes are there: carboxypeptidase, aminopeptidase, dipeptidase, and they are acting on the polypeptides to give the final product of amino acids. Enterokinase is helping for the activation of trypsinogen. Okay. After that, different enzymes like isomaltase, maltase, nucleotidase, equacidase are also in cross-border enzymes. About zymogens, we have talked about the zymogens. Generally, the digestive enzymes, they are being synthesized in their inactive form. Okay. The question is that why it becomes necessary to synthesize them in inactive form is that if they are synthesized in the active form, they may be able to digest the tissue that synthesize themselves. Okay, so to prevent that auto-catalytic of that cells or tissue by that enzyme, it becomes necessary for the certain enzymes to be synthesized in their inactive form. So this is generally the proteins, as you know, are being synthesized through the transcription translation process. So now, what gives the direction for that protein or enzymes to come out of that the cells that is secreting them? These are they have the N-terminal signal sequence. Okay, signal sequence means it has signal that gives direction. If it is being synthesized through the endoplasmic reticulum, rough endocrine, it comes towards the Golgi apparatus through the vesicles. That direction is provided by this N-terminal signal sequence. If that is lacking, the proteins may be synthesized inside the cell itself. Okay, that may not get entered towards the may not be secreted. Okay, so in this way, the synthesis of zymogens or any proteins may occur. And when zymogens they come to their site of action, they are activated by proteolytic cleavage. Okay, example of this zymogens: pepsinogen, trypsinogen, chymotrypsinogen, proelastase, prophospholipase. Right? For the activation of these zymogens, your example is one of these pepsinogen. They are synthesized in being their large molecules. Okay, and certain parts should be kept so that to expose the active site. Means when it is in its synthesized form, it should get maximum of that active site when it is in their synthesizing cells. But when it comes to the location where it should show its action, then it should be cleaved so that the active site get exposed and the whole enzyme is activated. Okay, so pepsinogens with the help of HCl, that catalysis occur. This is autocatalysis, example of autocatalysis. Right? Means they are cleaving that certain portion to expose the particle active sites of that enzyme. So to prevent the premature, to prevent the activation of trypsinogen to trypsin, if inside the cell, if it has occurred by error, there is one important enzyme, pancreatic secretory trypsin inhibitor, PSTI. Okay. And actually, the PSTI is inhibiting the trypsin if it is erroneously activated within the pancreas. If by error, if it gets activated, then PSTI molecule will inhibit that activated trypsin. For regulation of secretion, there is secretagogy. Okay, it is a substance that causes another substance to be secreted, right? So generally the neurotransmitters and these GI hormones are the secreted different. We have talked about individual gastrin, secretin, right? Cholecystokinin, GIP, motilin, and somatostatin under these secretagogue molecules. So after that, there is pancreatic secretions, bile secretions, okay, gastric secretions that is helping for the digestion and absorption in the GI tract, and that will be we'll discuss in next class. Okay. So if there is any problem, I think you can give me message over this chat box. GIP, gastric inhibited polypeptide is there, GIP. Generally, it is inhibiting this gastric acid secretion. Gastric stimulated release of gastric acid. Okay, for today's we'll end the class. So next continue will be in next classes.